Hey all, Pedantic Skeptic here, and as promised, I have a video about hybridization. So we're going to address the creationist questions about why we don't see hybrids and things like that. So I just wanted to preface that hybrids are not transitional species or forms. Hybrids are the result of the mixing of two already existent species that are in the same place at the same time. So I don't know why they ask this question, why we don't see hybrids, but we do see hybrids. So I have a presentation to show about hybridization. We're going to kind of get into it. It's not going to be an exhaustive or I don't want to say it's going to be overly comprehensive, but it's going to kind of cover some of the basics of hybridization and speciation and things like that so that people have that information. So let's do this. All right, so hybridization. Before we can talk about hybridization, we got to talk about what exactly a species is. So, the traditional definition is a population of distinct organisms that are capable of interbreeding with each other and bringing forth viable and fertile offspring. Now, that's important, but there are some issues with that definition. So, this original definition of species does exclude many hybrids because of the viable and fertile part. There are plenty of sterile hybrids between things, but one of the things to keep in mind is that not all organisms reproduce sexually, as well as the fact that genetic material can go between organisms through non-sexual means. Other issues would be the fact that sterile individuals can exist within populations, and so there can be an individual that was produced by a mating that, for one reason or another, generally because of an anomaly or an injury, is no longer able to actually reproduce. And so, by this definition, it would technically no longer be part of any species, which just sounds kind of ridiculous. It makes no sense. So I have kind of a different, more nuanced way of looking at what a species is. I would say a species is a group of organisms that has distinct unifying features that are reliably recognized by the trained eye and are differential from other groups of organisms that belong to other species. So that means that the feature has to be more or less unique to that particular group of organisms, and it has to be um, detectable enough where you can rule out other species and say definitively that it is part of this particular species. Um, these features should be more specific than genus or family level. And so the way that kind of works is if anybody's ever used a dichotomous key, um, there are features that unify a family. Um, so, for example, the family Saturniidae, it's a moth family. So, it's in the order Lepidoptera, which is within the Insecta. So, if we break this down, um, it's a eukaryote, and you know that because the cells have a nucleus and membrane-bound organelles. Um, it's an animal because not only do the cells lack a cell wall, the organism is a multicellular eukaryote. That's important because there are unicellular eukaryotes such as yeasts and protists, but it's a multicellular eukaryote with an internal digestive column and cell type specialization. Um, so those are broad definitions that encompass a lot of things. Um, it is a protostome, which is the fact that during blastulation, or gastrulation, the hole that forms into a hollow chamber, that first hole will become the mouth of the organism. Um, it's an ecdysozoan, which means it has a rigid external cuticle that must be shed. It is an arthropod because it is an ecdysozoan that has legs with joints to them. It is an insect because it is a Hexapod meaning it has 6 legs and it has external mouth parts. So not all hexapods have external mouth parts. It's kind of weird, but just roll with it. It's it's an insect because it's not only a hexapod with external mouth parts, it also has a pair of antennae. It is a lepidopteran because it is holometabolous, meaning it has complete metamorphosis, 4 distinct life cycle stages. It has 4 wings with scales over its wings and body. It is a moth, which all lepidopterans are technically moths. It is a saturniid because it has the features required to be a saturniid, which have mostly to do with wing venation. When you get to the lepidopterans, the patterns of wing venation are what mainly distinguish the families, as well as the presence or absence of a frenulum, which we're not going to talk about, but it's just a little hole on the wing to clip basically the other wing to it so that the forewings and hindwings are connected. Saturniids lack this, as well as Lasiocampids and several other families of lepidopterans. And so it is a saturniid. The mouth parts are vestigial, but that doesn't make it a saturniid. And then we can get into, more genus level classifications. So the genus Actius, for example, has a number of features. If you were to do like a drill down search and you have like a specimen of a moth and you figured out that it's a saturniid because you've looked at the wing venation, now you would look for other features that tell you it's in the genus Actius. Then what you would do is you would use the key to Actius to distinguish the features of the different Actius species. We should actually run through an example of that. Let's see. Okay. So this is how this would work. So let's just pretend we have a tree we're looking at, okay? Woody plants. The outer tissues are thickened and have bark and winter buds during the dormant season. We're going to click woody plants. Then you have your broad-leaved woody plants, and you have your needle-leaved woody plants. We'll just go with broad-leaved. Oh my god, shut up. Yes. Continue. Okay. So let's start with leaf type. Okay. Is it a compound or a simple leaf? Let's just say compound and apply. Leaves per node. That's going to tell us if the leaves are opposite or alternate. There's one leaf, which means they're alternate leaves. That's going to rule out some stuff. Leaf blade edges. I'm going to say the edge of the leaf blade has teeth. Leaf duration. Leaves drop off in winter. Armature on plants. No spines, prickles, or thorns. And so it just keeps narrowing things down. So let's just click on the Caria for a moment. I want to go to dichotomous key. Here's a dichotomous key. Plants reproducing by seeds. Plants producing true flowers. They're going to be dicots. Plants definitely woody. Leaves are going to be alternate. Inflorescence is not pseudanthale, fruit-various. Leaves compound. Is the perianth zygomorphic or actinomorphic? So zygomorphic is bilaterally symmetrical. Actinomorphic is going to be radially symmetrical. Think A for actinomorphic, it's all over the place. Leaves are going to be pinnately compound. Leaves, the fruit's not a legume. We're going to collect Juglandaceae, and then we're going to click Carya. So if you remember here, I clicked, okay. So, let's click Juglandaceae, so the family Juglandaceae. These are just some of this is an information about the Juglandaceae, and then within the Juglandaceae, you can select 2 different genera based on these particular characteristics. So, Juglands would be characterized by having leaves that have this number of leaflets. Middle lateral leaflets are the largest. The branchlets have a chambered pith instead of a solid pith. That's important. Drupe is indehiscent in the Juglands. It's a 4-valve dehiscent fruit in the Carya. And so if you know what the fuck all these terms mean, you can very easily figure out, okay, the genus Carya is defined by these characters, whereas, these characters here. Whereas the genus Juglands is going to be characterized by these. So we'll just click Carya. So here are the kind of some of the information for just the genus Carya. If it has all of these features, it's in the genus Carya. And remember that within the genus Carya, it's going to have all the features that make it a dicotyledonous. It has all the features that make it an angiosperm. Of course, it's a plant. It's a multicellular eukaryotic organism that has cell type specialization, a sterile lining around the gametes, and a few other things that make it a plant. It's going to be an angiosperm, which means it produces flowers that bear pollen and an ovule, and it produces a seed within an enclosed fruit. It's going to be a dicot because its seeds has 2 cotyledons and some other features that make it a dicot. We're not going to get into all that. And then it's a Juglandaceous plant, so it's in the family Juglandaceae. And then the genus Carya. None of these features mean that it's outside of the family Juglandaceae. None of these features violate what is required to be Juglandaceous, but these features make it Carya. And then we can have all of your, so Carya cordiformis is a Carya with a yellow terminal bud. What I will say, having worked with Carya cordiformis, is that sometimes during the growing season, the bud isn't all that yellow because it's still forming, but once the plant goes dormant, you can clearly tell it's this weird yellowy orange color. It's the only Carya that has that, so dichotomous tend to first rule out some of the more distinct species. Then you can choose this lead, and, you can just keep following the dichotomous key. So anyways, speciation is the process by which a population diverges into multiple species. You can either have sympatric speciation, where this process occurs even though the species are sharing a range. For one reason or another, they don't breed with each other anymore, and enough of a barrier forms, genetically speaking, to make them separate species. One thing that might happen is you may have one group utilizing a certain food resource or being active at a certain time of the day. You see that in a lot of moth genera, like the Calosamia, which we're going to talk about later. Allopatric speciation would be some sort of barrier forms. You see this in fish where like lakes get fragmented or things like that. So there's not a giant difference there. It can happen either way. We also have things called ring species. Doesn't necessarily mean that their ranges are in a ring per se, but it's kind of like where you have a set of species where there's a common ancestor um, that didn't get separated by any barrier, but it spread apart and speciated over time. And so you have species A and B sharing a range, species B and C sharing a range, species C and D sharing a range. So species A can hybridize or interbreed with species B very easily, but it can't interbreed with species C or D. Species B can hybridize with species A and C, but not with species D. Species C can hybridize with B and D, but not with A, and so on. And so this happens a lot with certain small vertebrates like salamanders and things where they can spread out over successive generations, but they're not so motile. They can, move back and forth across their natural range during their lifespans. So, just think of very small vertebrates that, kind of are ground dwellers. Salamanders are a really good example of ring species because they don't tend to move a lot during their life. But if you think over the span of, 500, 600 years, salamander populations can really take over, an area. Hybridization occurs when two different species mate and produce an offspring. Usually, the hybridization occurs between species that are in the same genus, but you can have intergeneric hybrids. You almost always see this only in plants. And, you have some instances where it can happen in, like the Lepidoptera. Generally, this produces very weak, sterile offspring compared to the plants. And we'll talk about some examples of this. But generally, intergeneric hybridization doesn't bear very many fruits, if you will. Here's where things can get a little bit confusing. When you breed two animals together, it's the male parent x female parent. So if it's a moth hybrid where it's Hyelaphorus cecropia male mating with a Columbia female, it's Hyelaphorus cecropia x Columbia. Whereas with like an orchid, for example, it's a plant. So the parent that made the seed comes first. So if you pollinate Cypripedium macaulay with Cypripedium reginae, it's Cypripedium macaulay x reginae, not reginae x macaulay. It can be really tricky to just remember that rule. And, I have my own opinions about that I'm not going to get into because they're not relevant, but just remember that animals and plants are kind of different in how we describe them. Now we need to get into the barriers to hybridization, because hybridization doesn't occur just willy-nilly all over the goddamn place. We have some barriers, some things that prevent hybridization from happening. There are prezygotic barriers and then postzygotic barriers. So things that prevent that fertilization from happening in the first place, and then things that happen after that fertilization event to prevent the hybrid from actually being a thing. Now there's a caveat here. We're going to talk mostly about natural situations. So if you look on the other side of the equal sign, this monstrosity, and I thank my Christian friend for pointing this out to me, is Phlebosia. It's an intergeneric fern hybrid between Phlebodium, which is the middle picture, that's the blue star fern. I love Phlebodium aureum, it's a nice fern, and Pyrosia. Now, Phlebodium is a North and Central American genus of ferns, whereas Pyrosia is an Asian and Australian genus of ferns. They would never meet each other in nature, but if you grow their gametophytes closely enough together, you can get this intergeneric mess of a plant that is a hybrid between the two. It certainly looks more like Phlebodium than Pyrosia, but it's just a fucking disaster. Just get a Phlebodium. Jesus Christ, people. So, let's talk first about differences in the recognition of suitable mates. Some organisms aren't going to recognize other organisms in general as a potential mate. Okay? The logistics of pairing them isn't going to permit the transfer of gametes. So, one example is this giraffe and this frog. It wouldn't end well for the frog if a male giraffe tried to mate with it. And that's even fucking assuming that the giraffe would want to mate with the frog in the first place. I seriously doubt it. There are other problems, like frogs fertilize eggs externally for the most part. Giraffes fertilize eggs internally. And before we continue, just keep in mind that these barriers aren't mutually exclusive. So we're going to talk about some other barriers like gamete chemistry being different. If you take frog sperm and put out a giraffe egg in the lab, you're not going to get a fertilization event because the sperm and egg are just so different. So, even if a giraffe managed to, get its sperm on a frog egg without killing the frog in the process, or if a frog managed to get its sperm inside of a female giraffe by some miracle or freak accident of nature, then, fertilization still wouldn't happen. But, there are a number of things here that we got to talk about. And so let's just forget the idea of a giraffe trying to mate with a frog for a moment and, forget about that stupid YouTube video of the monkey or chimp using the frog as a sex toy. That's not necessarily a reproductive activity, but, to each their own. Please don't harm frogs. Thank you. Anyway, the different species often use different ways of attracting mates. So, crickets, for example, chirp to attract mates. Frogs croak to attract mates. Something that uses olfactory cues, like a moth, where the female releases a smell to attract the males, isn't going to cue in on a frog making noise as, hey, come mate with me. It's going to just either ignore it or be freaked the fuck out. Meanwhile, the frog probably won't even smell the moth pheromone, things like that. Moths of different species probably won't recognize the smells of other moths as the pheromones of their to mate with. So one of the things people do when they make hybrids of moths in captivity is they will confine a female of species A to a cage next to a cage with the male of species A and the female of species B. So the male smells the female really close, and when he bumps into the female species B, he just simply thinks that that is the female species A in the first place. This is shit that probably wouldn't happen out in nature, but because humans like to fuck around with things, we can oftentimes make this happen. So, again, there are differences in recognition. In angiosperms, I should have specified that where I said in plants, because not every plant's an angiosperm, but pollination biology. Pollinators don't oftentimes visit multiple species of plants at the same time. There are some pollinating insects where they will visit one plant species and only one plant species during a single foraging trip or for their whole lives. And so if you have plant A, and we're assuming that they're, these are all insect pollinated, not wind pollinated and things like that, but there are, it's going to be a lot harder for pollen from an insect pollinated plant to get onto a wind pollinated plant. Maybe not as hard for wind pollinated pollen to get onto an insect pollinated plant, but it probably wouldn't do much. Now, if, an insect isn't attracted to plant B, but is attracted to plant A, it's not going to put pollen from plant A onto plant B. We also have differences in range. If they can't reach one another, they're not going to be able to mate. And it's as simple as that. And we're going to talk as we get towards the end of this presentation about how that's changing. But just remember that If two species, you have a lot of frog species in Europe that look like a lot of frog species in America. Like the European tree frog looks a hell of a lot like the gray tree frog, and the European pond frog looks a hell of a lot like the wood frog. Um, and maybe that's an example of convergent evolution or something like that. But they would never get close enough to one another in the natural settings to actually be able to breed with each other. Here's another one. Differences in breeding time. So let's first start with angiosperms because, I don't want this to all be about animals. I don't want this to all be about plants. I want to have a good mix of both. Now, if plant A blooms in, April and plant B blooms in July, their gametes aren't going to cross paths. Okay? Um, you have some plants that are precocious bloomers, like a lot of the cherries, for example, within the genus Prunus. Prunus is a big fucking genus, but within the genus Prunus, you have some species that bloom right when it gets warm, and their flowers come out before their leaves. And then you have some species that bloom after the leaves come out a little bit later in the season. They're not going to necessarily overlap in their bloom time enough to have crosses happen. Now, that doesn't mean it never happens, but in general, if they're blooming at different times of the year, they're not going to pollinate each other. Other things could be animals that breed at different times of the year. So, for example, spotted salamanders and marbled salamanders are both in the genus Ambystoma. Ambystoma are one of the few vertebrate genera where hybridization between individuals can result in fertile offspring. They're not the only, but they're one of only a few that we know of. A lot of times when vertebrates, especially mammals, and I'm not saying salamanders are mammals, but often when vertebrates hybridize with each other, you get sterile individuals from that. A good example would be the mule, which is a donkey-horse hybrid. I forget the direction of the hybrid, and I believe that we'll talk about the direction mattering later, but I think that in some cases, if one of them is the father, sometimes the males can be fertile, but we'll talk about that later. But for example, spotted salamanders and marbled salamanders breed at vastly different times of the year. The spotted salamander breeds in temporary pools of water right when the ground thaws, early spring. Well, I shouldn't say right when the ground thaws. The ground thaws and it rains a couple times, then they migrate to their breeding pools and do their thing. But marble salamanders wait until the summertime, where they migrate to their breeding pools, mate, lay eggs when the vernal pools are dried up, and they basically will sit on the eggs and pee on them or sweat on them to keep them moist enough, because the female salamander can burrow herself into the ground where she can soak up some water and then come back up to the surface and wet the eggs. And she'll do that until the rains in the fall fill that vernal pool back up with water, at which point she will leave. Her eggs will hatch soon after and sit in the vernal pool when it's frozen over. So a marble salamander and a spotted salamander would never be able to breed with each other. Now, axolotls, for example, are an ambystoma that you can easily hybridize with a number of other ambystoma because they breed kind of at any time of the year, as long as the female is receptive. And so you can throw a male salamander within the genus Ambystoma in with her if he's in, breeding mode, and he'll breed with her. I don't recommend doing that, but you can if you really were determined to. So differences in breeding time will make a big difference in whether or not two things will actually hybridize with each other. And some of these people have found that this is a weaker barrier because you can fuck around with the animal or plant's phenology, or with plants especially, you can actually collect pollen and refrigerate it for quite a while and it will stay viable, and then you can pollinate a later flowering plant and it will produce perfectly fine seeds. People who grow pitcher plants and some other things, some other ornamentals, will do that. Okay? Now we need to talk about differences in gamete chemistry. This is our final prezygotic barrier. The sperm has to be able to penetrate the egg. Now, we're excluding fungi here, because fungi don't really have sperm and egg. Fungi are weird because they have multiple sexes, and there are clusters of sexes that are able to mate with each other, and as long as they're compatible on a cellular level. But it's kind of the same principle where there have to be matching receptors and recognition sites on the cell wall, or yeah, fungus has a cell wall, but animals don't. But the external covering of the cell, the membrane or whatever it is, that allow that fusion of the gametes to occur. If that gamete isn't recognized by the other gamete, fertilization just doesn't happen. This is also where we talk about kleptogamy, which you see in some animals and plants. Dandelion is a good example in plants. So basically what happens is when those gametes meet, even if they don't fuse, there can be chemical reactions that stimulate that cell to undergo divisions and whatnot. In animals, sometimes the sperm of a male can trigger ovulation and other things. So kleptogamy is basically where an organism takes the gametes from another organism, but doesn't incorporate the genetic material into its offspring. So salamanders, for example, will sometimes pick up male spermatophores. So the way salamanders mate is kind of weird. The male makes a big packet of sperm called a spermatophore. That itself isn't weird because most insects use spermatophores, but he'll make a spermatophore and put it somewhere for the female to then use her back end to pick up and kind of just shove into herself. Sometimes the female doesn't fertilize her eggs with that sperm, but that sperm triggers her body to release the eggs so she can lay them. That's called kleptogamy. Dandelion also does this, where the pollen will hit the flower, but it won't fertilize the embryo inside. The plant will just spontaneously make an embryo that's a clone of itself, and then that seed will have a clone of the mother plant, but it still requires that sperm or the pollen to hit it and basically kind of unlock that seed production mode, if you will. Of course, there's a series of biochemical processes that occur. I'm telling you this in very simple terms. If you're interested in it, you can look it up. It's really cool stuff. So those sorts of things happen. It's really interesting shit, but I don't want to overcomplicate things. What was I gonna say? There was one other thing. Just give me a sec. Um, let's see. We had salamanders, the sperm. Oh yeah. Some parthenogenic lizards. I'm sure everybody's heard of like the lesbian lizard thing, where like parthenogenic whiptail lizards still like take turns humping each other because that I think it's just like the pressure being applied to the female in that certain way triggers the ovulation and things like that. But there's no actual movement of gametes between the individuals. Uh, really interesting stuff. So now we're going to get into some post-zygotic barriers. Reduced hybrid viability is where those hybrid offspring just don't do very well. Maybe the fertilization happens, but that embryo dies very shortly after fertilization. This can often be the result of mismatches in chromosome numbers between the individuals. Sometimes what happens is the diploid number is such where the haploid number is an odd number, or the parent species differs so much in how many chromosomes they each had that the offspring is unable to actually undergo cellular replication. This generally affects fertility more than viability. Viability is just the ability to survive. You can be sterile and otherwise healthy. If you're sterile, you're just unable to reproduce. And sterility is, a gradient type of thing, and we're going to talk about that later. But, if you're not viable, you're not surviving in the first place. So, hybrids sometimes have some issues. And we're going to talk about why that is in just a moment. Now, in plants, sometimes hybrid embryos are aborted. So here's where we get into some story time. Does anybody here know what embryo rescue is? That's okay, because I'm about to fucking tell you. So, for those of you who don't know what tissue culture is, and this is important, embryo rescue is not something that happens in the fucking wild. Embryo rescue only happens in the context of human activity. Embryo rescue is the process of taking a plant hybrid embryo. So, you have a flower where you put pollen from another. And right now, we really only deal with angiosperms because, like the things inside of like ferns and their gametophytes right now are just too tiny for us to really fuck around with. But in any case, you take the pollen from species A and put it on the flower species B. And what's going to happen, what we know is that when that happens, the seed starts to form, but the plant eventually recognizes, hey, you're not like me, I'm going to kill you. And the plant basically stops putting nutrients into that developing embryo inside the seed, and the seed pod withers away and falls off, and the embryo dies. It's, plants can give themselves abortions, which is really cool. But anyway, what embryo rescue is a person saying, before this happens, I'm going to cut out that embryo from that plant, put it on a tissue culture medium, which is basically a nutrient medium that's usually solidified with agar. It's kind of like sugar water with fertilizer and other nutrients added to it. And by doing that, you prevent that embryo from dying, and it can grow into an actual hybrid plant. So that's what embryo rescue is. And right now, people are using this method for ash and chestnut. So here's where story time comes in. We'll talk about ash first. Emerald ash borer is an invasive buprestid beetle that is killing off native ash species in the United States. Okay? It's kind of all over the goddamn place. It just hit Rhode Island a couple years ago. So it's all the way out to the east. And, it's in Colorado, and it's, pretty much everywhere now. And the problem is that emerald ash borer causes so much damage to the ash tree when it bores through the cabium and the other parts of the bark that the plant doesn't know what to do. It stresses the fuck out and dies. Now, in plant-insect interactions, there's a concept called resistance and there's a concept called tolerance. Resistance is not getting attacked by the pest in the first place, and tolerance is being able to deal with the damage that the pest does to you, because you can just say, oh, I lost a branch, let me just grow that branch back. And the native ash trees in the United States have no idea what the emerald ash borer is, because they've never co-evolved with anything like it, and so that's why they die. But the emerald ash borer, in its native range in Asia, lives with several ash species, including Manchurian ash and a few other species that don't die when the ash borer goes after it. So what people are trying to do is they're trying to create hybrids between Manchurian ash and our native ashes, as well as some other native Asian ashes. And the hybridizations almost always fail. And so one of the things you can do is you can do embryo rescue. Not saying ash is particularly easy to tissue culture. It's very difficult. Ash is not an easy plant to work with. And I'm not going to get into all the nitty-gritties about ash, but ash is not easy to work with. But that's one application. The other application is the American chestnut, because Asian chestnuts have co-evolved with a fungus called chestnut blight that kills American chestnut very slowly, And so they're trying to cross American chestnut with some Asian chestnut species in order to try and produce the chestnut blight resistant or tolerant American chestnut hybrids that retain the American chestnut characteristics while having that resistance. They're trying to basically put the resistance genes into the American chestnut while saving some of the American chestnut qualities that we know and love. And the reason why we haven't bioteched the shit out of this yet, where we use CRISPR to selectively cut and paste the genes, we just don't know enough about the genome of these native chestnuts to know what genes are responsible for conferring this resistance to the fungus. So the next best thing is to just play around with hybridization. But they don't like being hybridized very much, especially the Asian and American species, so embryo rescue is an application for that as well. Now, tangent aside, you can have animals where the eggs fail to hatch, or it's stillbirth or something like that. And here's where we get into the direction of the cross mattering. Especially with invertebrates, which species you designate as the mother species can make a big difference in how well that embryo does. In Lepidopterans, sometimes the species that produces the bigger egg gives you better outcomes for embryo survival. Sometimes the species that's more of a generalist in how it eats or how it lives is going to be the better bet. And it's going to be species, but it's going to be a case-by-case basis. It's going to differ, and there's a lot of unknowns here. It's really cool stuff, but what you're going to have to realize is that because of the complexity of life as we know it, you're going to need to, try crossing it both ways. You have species A and species B, cross a male of A with a female of B, cross a female of A with a male of B, see what the fuck happens. Then you have reduced hybrid fertility. The hybrids survive and are perfectly healthy, but they're sterile. Now, sterility can be from a couple of different mechanisms. Sometimes something is sterile because it doesn't have gonads. Sometimes something is sterile because it doesn't produce gametes. Sometimes the gametes are unable to fuse with other gametes. Okay? So sterility isn't a one-size-fits-all, but the fertility of those embryo offspring are oftentimes compromised by being hybrids. Again, the direction of the cross can be important, and I'm not going to list all the examples that I know, like from the Lepidoptera and, from some mammal stuff, where, if you use species A as the male, you may get some fertile offspring, but if species A is the female, you're not going to get any fertile, that doesn't really matter here. But just know that sometimes the parent who is the father or the parent who is the mother is going to make a difference in whether or not those offspring are able to reproduce themselves. And sometimes the males are fertile while the females are sterile. I think that just has more to do with how spermatogenesis differs from oogenesis. We're not going to get all into that because it's just a very complicated topic that I have limited knowledge on. But a lot of times, especially with moth hybrids, the males are fertile but the females are sterile for whatever reason. Now we get into something called hybrid blend zones. This is studied mostly with Lepidoptera, but you can have it occur with a lot of other species as well. So this is looking specifically at one group of organisms, the tiger swallowtails. They're fun organisms to work with. A tiger swallowtail is a specific group of swallowtails. The genus Papilio is pretty big. Okay, it's like the biggest genus in the Papilionidae. It's a big genus. And one group is the tiger swallowtail group that encompasses several species. They put Batis phyllenor on this as an outgroup for whatever reason. I think it just gets a mimicry. But what happens, you see this a lot with leps, is you get two species that are distinct, that have different ranges, but you then have areas where their ranges overlap a little bit. Like you can see here, Glaucus and Appalachianus have, range overlap, as well as Glaucus and Canadensis. And so where the two species meet, you get a hybrid blend zone, where you're going to see both species as well as hybrids between the two species. And you'll see different hybrids. It's really fucking cool because you'll get like first generation Appalachianus x Glaucus. But then you might get back crosses in either direction. You might get like third and fourth generation hybrids. And so you get something that may look almost entirely like Glaucus, but it has something kind of funky about it. So maybe several generations back, there was an Appalachianus involved. Or you get something that's like exactly halfway between the two species. And so you can get these hybrid blend zones where you're seeing things that don't quite match the characteristics of one species, and it's probably just a hybrid to some degree. And then we get into hybridogenesis. If two species are close enough, they can actually produce a third fertile species through hybridization. So with these moths, you have the big picture is Hylophora casluensis. It is from a specific area in California where Hylophora glaveri, which is the top little picture, and Hylophora murialis, which is the bottom picture, mate with one another. Now, all three of these moths in these pictures are male, so obviously, I mean, I'm not saying that there can't. I've seen male Luna moths mate with each other before, but that wouldn't actually produce an offspring. But to each their own. We love gay moths over here. But you also see that with some frogs and a number of other species. Where you got a third species because of the mating activities of two other species. So now let's just quickly go through some examples of hybrids. When people rear Saturniid moths especially, they really like to fuck around and cross different species of Saturniids. They're curious as to what might happen. And one of the biggest reasons why they also do this with Papilionids. One of the biggest reasons that people do this is because you can actually hand pair a lot of leps, especially the bigger ones like the Saturniids that are, several inches across, where you grab the male and, you either gently squeeze his abdomen or sometimes you can just hold him upside down for long enough and he has these claspers and you can manipulate the abdomen to grab onto the abdomen of a female, and as long as he can get that sperm inside of her and doesn't harm her in the process, which can happen, you can actually have the male's genitals puncture the female's body and kill her or prevent her from laying eggs. It's happened. There are a lot of there's a lot of weird shit that can happen, but what can often happen is people can manipulate these moths to mate with one another, even if they're separate species. Calissae and Hylophora are very oftentimes used. The three Calissamia species are kind of weird because they use the exact same chemical to attract mates, but they just mate at different times of the day. So Calissamia securifera, which is found very far south compared to the other two species, mates, I believe, around noon to 2 or 3 in the afternoon. Promethea, which is the most widespread of the genus, mates just before sunset, whereas Angulifera mates between 10 p.m. and midnight. If you take a female Promethea in the evening when she's calling, releasing her pheromones to attract a mate, and you keep a light over her, and then it gets dark, you can actually keep her producing those pheromones and attract a male Angulifera to her. Artificial lighting might be to blame for some of these accidental pairings. One year I accidentally reared hybrids between Angulifera and Promethea because somebody had caught a wild Angulifera female, and when the offspring produced their cocoons that had Promethea traits, I realized I had a hybrid. But you can hybridize the Calissamia quite readily. Sometimes if the generalist species is the mother, so if Promethea is the mother, the offspring do better than if the more specialized species, and I'm talking dietary specialized versus generalized, but there are other ways of being generalized and specialized. But anyway, sometimes if the mother's the generalist feeder, because of the way that cytoplasm in the egg works, you have more viable offspring. They can eat better and they recognize food more easily, so they're actually able to eat when they hatch and they don't fucking starve to death. But anyway, people play around with a lot of these big fucking moths to see if they can get them to hybridize with each other. we also do this with plants, especially orchids. Orchids are grouped into alliances like the Cattleya Alliance, Oncidium Alliance, Dendrobium Alliance, Vanda Alliance, and some other things. And generally they do correlate quite tightly with actual taxonomy of the Orchidaceae, not always, but quite often. And you can actually do a lot of intergeneric hybridization between the orchids. And I had an argument with somebody about whether or not, everything in the genus Cattleya is technically the same species because it's capable of interbreeding. And again, it depends on what you define as species. But the main thing here is that we recognize these different genera of orchids, yet we can still breed them together and produce perfectly viable, fertile hybrids. And if you look after the Cattleya, you have your grocery store orchid, that Phalaenopsis. That big-ass, fat-petaled Phalaenopsis is different from wild-type pure species Phalaenopsis that you see below it with the falcate tiny little petals and the small flowers. Repetitive hybridization of Phalaenopsis just makes it look like that ugly fucking thing up there. I'm not a big fan of Phalaenopsis. I'm a fan of Dendrobium, Cypripedium, Cymbidium, Oncidium, and Cattleya, and their allied plants, as well as some other really cool orchids. Not a big fan of the Phalaenopsis. I call them the normie orchid, but, that is kind of the extent of most people's knowledge on orchids. But you can make intergeneric orchid hybrids. And finally, let's talk about hybrids that are a result of climate change and human activity. So, in mammals, we have coy wolves and growler bears. Because of climate change and humans pushing things out of their native ranges, when coyotes and wolves and then polar bears and grizzly bears, kind of meet up because their ranges are being shifted by climate change and things like that, they can produce hybrids. So, a horny wolf doesn't want to kill a coyote, it wants to fuck a coyote, and then it makes a fucking hybrid. Same thing with the polar bear and the grizzly bear. I don't want to know the logistics of all that. And then a lot of the willows in the United States nowadays are the result of invasive willows coming in and hybridizing with the native willows. Sometimes you get a dilution of native genes by these hybrids. It can be a real fucking problem. So, that is my presentation on hybrids. So yes, to the creationists, we do see fucking hybrids. Checkmate.